So let's start with metaphase now. So meta is second. Meta is second. Phase is nothing but stage. So this is the second stage of the karyokinesis of mitosis. So let's see what happens during this metaphase inside the cell. So during metaphase, that is in the early metaphase. Now let's see early metaphase. In the early metaphase, chromosomes, condensation of chromosomes is completed. So in this metaphase, chromosomes exhibit maximum condensation. So chromosomes show condensation. Chromosomes exhibit maximum condensation. Maximum condensation. So this metaphase is the best stage to study, best phase to study the number of chromosomes and to study the morphology of chromosomes. So this is the best phase to study the number of chromosomes. The best phase to study the number of chromosomes. Why? Because they are showing maximum condensation. Maximum condensation so that we can observe these chromosomes very clearly under the microscope. So this is the best phase to study the number of chromosomes, to study the number and morphology of chromosomes. Morphology of chromosomes. Okay. So in the early metaphase itself, that is at the beginning of the metaphase, nuclear membrane undergoes complete disintegration. So nuclear membrane is completely disintegrated by early metaphase. Completely disintegrated by early metaphase. Actually, by the end of prophase, what happens? Nuclear membrane starts disintegrating, and by the early metaphase, that is by the beginning of metaphase, this nuclear membrane is completely disintegrated. Next thing is, by the early metaphase, spindle apparatus formation is completed. Spindle apparatus formation is completed. Actually, by the late prophase, what happens by late prophase? Spindle apparatus formation is initiated by late prophase, whereas its formation is completed by early metaphase. So after formation of spindle apparatus, the spindle fibers present in spindle apparatus attach with the chromosomes at the point of kinetochores. And then after attachment, they bring all these chromosomes, spindle fibers bring all these chromosomes towards the center of the cell. So the movement of chromosomes towards the center of the cell is called as congression. So spindle fibers of spindle apparatus attached with chromosomes at kinetochores. So after attachment, they bring these chromosomes towards the center of the cell. So the movement of chromosomes towards the center of the cell, so this movement is taking place in the late metaphase. So up to early metaphase, attachment of spindle fibers with chromosomes to kinetochores. Now we are going to study about late metaphase. So what happens in late metaphase? Now till here early metaphase. Now let's see about late metaphase. So in late metaphase, what happens? These spindle fibers are bringing the chromosomes towards the center of the cell. So this movement of chromosomes towards the center of the cell is called as congression. So movement of chromosomes towards the center of the cell is called as congression. Whereas alignment of chromosomes at the center of the cell is called as metaphasic plate or equatorial plate. Alignment of chromosomes at the center of the cell is called as equatorial plate or metaphasic plate. equatorial or metaphasic plate. Metaphasic plate. So let's see how this happens. So I explain you these steps with the help of diagram. So this is the animal cell membrane. This is the animal cell membrane. And this is centrosome. These are centrosomes. And these are centrioles, which are arranged perpendicular to each other. These are astral rays, that is microtubule fibers. Then these are, I am making only 2 chromosomes. These are the 2 chromosomes which are brought towards the center of the cell. So this is centromere. Now kinetochores. So these are the spindle fibers. Spindle fibers. There are 2 types of spindle fibers. Now let's study the type of spindle fibers. There are 2 types of spindle fibers. So this is the alignment of chromosomes at the center of the cell to form equatorial plate or metaphasic plate. Okay, now let's study types of spindle fibers present in the spindle apparatus. So per one cell, how many spindle apparatus are formed? During mitosis, only one spindle apparatus is formed. So spindle fibers. There are 2 types of spindle fibers. Spindle fibers. They are continuous spindle fibers, also called as pole-to-pole spindle fibers. Pole-to-pole spindle fibers. Then what are these continuous spindle fibers? These are continuously running from one pole to the other pole. These are called continuous spindle fibers. Spindle fibers. Okay. They are not attached with the chromosomes. Next type of spindle fibers. Next type are discontinuous spindle fibers. Discontinuous, or also called as chromosomal fibers. Chromosomal spindle fibers, or also called as tractile spindle fibers. Tractile spindle fibers. So these are the spindle fibers which are attached with the chromosomes. These are discontinuous. They are not running continuously from one pole to the other pole because they are attached with the chromosomes. These spindle fibers only bring the chromosomes towards the center of the cell to form metaphasic plate or equatorial plate. So this movement of chromosomes is called as congression, whereas alignment of chromosomes at the center of the cell is called as metaphasic plate or equatorial plate. So this is all about metaphase. Next, metaphase is followed by anaphase. So during anaphase, that is in the early anaphase, what happens? Centromere splits. After splitting of centromere of these chromosomes, chromatids of these chromosomes get separated and they start moving towards 2 opposite poles of the cell. This chromatid moves towards this pole and this chromatid start moving towards this pole. Okay. So let's see about anaphase.